Stephan Grupp, MD, PhD. He's the Director of Translational Research for the Center for Childhood Cancer Research at Children's Hospital of Philadelphia, better known as CHOP, and the Yetta Deitch Novotny Professor of Pediatrics at the University of Pennsylvania. He's a member of the Division of Oncology, the Stem Cell Transplant Group, and is a medical director of the Stem Cell Laboratory. Dr. Grupp graduated from the University of Cincinnati, completing his MD and PhD. His PhD is in immunology. He completed a pediatric residency at the Boston Children's Hospital, followed by a fellowship in pediatric hematology and oncology at the Dana-Farber Cancer Institute, and did his postdoctoral work in immunology at Harvard University. Soon afterward, he joined the faculty at Harvard until 1996 when he came to CHOP. His primary area of clinical research is the use of engineered cell therapies in high-risk pediatric cancers. His primary laboratory interest is the molecular control of leukemic cell growth and the development of new cell therapy treatments for pediatric cancers. Dr. Grupp is a reviewer for several journals and the author of over 120 peer-reviewed journal articles. He's also the National Chair for Stem Cell Transplantation for the Children's Oncology Group. We're excited to have you with us today, Dr. Grupp.
Okay, thank you very much for that very kind introduction and for the opportunity to present a little bit of data and maybe a little bit of speculation and try to give at least a partial answer to are we there yet? I will try to give you a sense for what engineered T-cell therapy is all about right now. And I think that in the end, I hope to convince you that we're almost there, not quite there yet in terms of solid tumors, but in terms of the work we've done in leukemia, we at least have a clear idea of what it looks like when it works. So we'll try to talk about that. I think it's very important to understand as I talk about all of this that this is brand new information. These are clinical have been underway only for a couple of years. We're just at the beginning of understanding how this might work, but we're also super excited about what we're learning right now. And as is always the case when we do this sort of work, it's just amazing that we have families and kids who are willing to partner with us in doing this very important research that we hope will make things better for kids with cancer. So let me just start by introducing some thoughts about where we are. So I think there are three waves of innovation in medicine over the last 150 years or so. And as with everything else it seems like these days, everything's coming faster and faster. So the first wave in medicine was the sense that there were actually chemical compounds that existed in nature or that you could synthetically make that would actually treat disease. And the antibiotic revolution is a great example of that. The discovery of penicillin as the first antibiotic, and then all the antibiotics that followed. Just extraordinary thought that you could use chemicals to actually modify disease processes and actually cure folks. This is also not too much after penicillin. It was the beginning of the chemotherapy era, and we were developing drugs that could treat cancer, and of course are the mainstays of cancer treatment now, these chemical blockers. But the issue in cancer, as any parent who's had to deal with this in their family understands, is that the side effects of chemotherapy are really, really substantial. So chemotherapy, what chemotherapy isn't is very targetable. We have made tremendous strides in both the use of chemotherapy and in the ability to deliver it as safely as possible to kids. It's still really hard, but I think what we've found out is that there are a lot of circumstances where we can really help folks, but there's some where pushing chemotherapy is really not going to change the standard of care. I think it's fair to say in 2015 that we've taken the chemo revolution as far as it's going to go. Now, after the chemo revolution, when I was in med school, we were beginning to see this idea that you could use synthetic and natural proteins as biological therapy. And the example, and I'll give you several examples of this in a second, are the antibody-type therapies. And so that has really been a second revolution in general medicine, but especially in cancer care, where this has really been targeted. In general, the use of these sorts of treatments are associated with much lower side effects, not always, but in general. They're drugs. You have to give them over and over again, and it's the nature of modern medicine that these things are extraordinarily expensive. What I want to talk about today is the third wave, and the third wave is really engineered cell therapy. There are a variety of immunotherapies, but my focus today is really sort of the ultimate personalized medicine, where we take patients' cells and engineer them to go after their own cancers. These are also therapies that are highly targeted. These are also therapies that have different side effects, that sometimes are substantial and sometimes are mild. These are also therapies that I have no idea how expensive they're going to be. You imagine they probably will be expensive, but there is a hope that if we make these cells right, they might stick around for a very long time, months to years. And what our real hope is that this might be something that you only have to do once instead of over and over again like chemotherapy. Now, if we go back to the second wave and think about examples, there are, I think, a number of different ways that people have made this work and work extremely well. And so the first drug is probably the most widely used biological agent for cancer, which is this drug called Rituxan, which is used for non-Hodgkin's lymphoma. It's an antibody. It is a chemical produced by your body, a protein produced by your body that recognizes a cancer cell and then causes an immune reaction which destroys the cancer cell. You can fancy that idea up, and you can take an antibody that binds to a cancer cell, and you can hook a toxin to it, something that would actually cause the cancer cell to die if the antibody brings the toxin to the antibody. There are a lot of examples of that, many of them, but inotuzumab, and I apologize for these names, they're terrible names, but inotuzumab is an example of that, and that's used in leukemia, and hasn't been approved yet, but it looks pretty powerful. And then you can take that idea another step further. You can actually use an antibody to hook up with a cancer cell, but on the other end of the antibody, you can have it also connect to a T cell. And T cells are cells of the immune system that can kill cancer cells, and that's what we're going to be talking about today. But this is a different way to do it, just using an antibody to hook the T cell and the cancer cell together and to cause a therapeutic effect. And so this is the first in its class of this kind of approach. It's very exciting. And that was just approved by the FDA for patients with leukemia. Now, for patients dealing with neuroblastoma, there's familiarity with this antibody called GD2 or 1418, and there's also another version that's used by Sloan Kettering called 3F8. And none of those, those get kind of technical, but they may be familiar to many of you. This is an antibody, or there are varieties of these antibodies that recognize this particular target on neuroblastoma cells. And the most successful protocols which have used this antibody have not just used the antibody by itself, but have also used it with other drugs that boost the immune system, drugs like interleukin-2 or IL-2, which is a T cell and natural killer cell activating protein, and an immune system booster called GM-CSF. So you probably need all those things to get it to work. Won't be talking today about vaccines, but there's been years and years of development in vaccines. In the cancer field, vaccines have been a challenge, and the reason why is that one of the things that cancer is awesome at is ducking the immune system. So cancer learns to hide from the immune system. It's very hard to overcome the fact that your immune system doesn't recognize your cancer. So there are a lot of vaccine programs out there. We still have a lot of hope for that, but there haven't been a ton of really obvious examples of success that lead us to say, okay, this is what it looks like, and this is what we ought to manage, and what we ought to model and ought to make happen. So let's talk about our third wave here. And our third wave is cellular immunotherapy. So we're talking about engineered T-cell therapy. We have to take cells from each individual patient, and I'll show you how this works. We have to overcome the fact that your T cells don't see your cancer by genetically engineering them, and I'll show you how we do that. And so we basically want to force the T cells to recognize. And you can see in that picture off on the right the interaction of a genetically modified T cell in orange that's going after a cancer cell. And I'll actually show you a movie in a minute of how that looks. So we want to genetically modify T cells to go after cancer cells. And this is an idea that has been around for 20 years, but it's only been very recently that we have kind of gotten the pieces together to actually show how this might work. And I think that that's why everybody is so very excited about this particular approach right now. So this complicated picture shows us how we genetically engineer these cells. So what you see in this picture on the top is a tumor cell. The tumor cell has a yellow target. You can pick targets. For neuroblastoma, it would be GD2. For leukemia, which I'll talk a lot about right now, it's this thing called CD19. It's just something that's on the tumor cell that you could recognize. On the bottom, you see a T cell, and the T cell has been genetically engineered with a protein. This is very cutting-edge stuff. And the protein is something that doesn't exist in nature. It's basically assembled out of other proteins. So if you look at that, what you're seeing on above the lining of the cell, that green-blue thing, is something that actually forces the T cell to bind to the target on the cancer cell. And on the inside, the blue, purple, red, are various pieces of proteins that turn T cells on. And we know a lot about the specific steps that have to go on in activating T cells so they actually can kill cancer cells. And we had to get these steps exactly right before this kind of approach would work. So that idea is one of the major steps forward in engineered T cell therapy, and it's called a chimeric antigen receptor, or a CAR. So I'll just call it a CAR from now on. It's this way of genetically engineering the T cells. So again, the CAR idea, early 90s is when this came up, and it was quite a while before we really started seeing this work. And the reasons that we really had to sort of push making these systems to work well is that we were limited by two things. The first thing we were limited by is that after you put the cells in the body, what you really want to have happen is these genetically modified T cells need to grow and divide, and you need to start with a small number of T cells and go to a very large number of T cells. That's how T cells work in your body naturally. If a patient gets chickenpox, if a patient gets chickenpox and their T cells have to eliminate the chickenpox, the T cells have to grow before the T cells will successfully eliminate the chickenpox. So that's expansion. The cells have to grow in the patient. There were a lot of clinical trials where they made awesome cells and they killed cancer cells in the test tube, but they didn't expand in the patient. Had to get past that. And then beyond that, we want something even better. We want something called persistence. And persistence means the T cells stick around. So in people, if they get chickenpox, the T cells that recognize chickenpox stick around probably for your whole life. And those T cells are on the hunt in case chickenpox comes back again. So that persistence is an extra layer of power of the immune system against cancer, because the idea is that expansion, growing these cells, will get on top of the cancer and destroy the cancer cells, but then the persistent cells can try to limit the risk of relapse. That you need both of those things for this to really work. And you know what I would say is that in the last couple of years we've actually seen this work. So let me show you a couple of movies on how this works. So I'll have to drag the movie in because the slide is not cooperating the way it should be. So what I'm showing you here, the big orange blob is a pediatric leukemia cell. And then you'll see a blue cell come in from the top, and that's a genetically engineered human T cell, actually a T cell that belonged to somebody who worked in my lab. And so you see the blue cell come in, and you see the blue cell connect with this cancer cell. And if you see this formation of a green rim, the green rim is actually the CAR protein, is this genetically engineered protein that is forcing this incredibly tight interaction between the T cell and the cancer cell. So that T cell sees the cancer cell. There's no hiding. So we're on top of that. We can actually make the T cell see the cancer cell. But that's not good enough. We got to go the next step. And so this is what the next step looks like. So now the green cells, let me run this all the way back to the beginning. The green cells are cancer cells, and the gray cell coming up from the bottom is an engineered T cell, human T cell. This is actually a leukemia that was derived from a patient who unfortunately died of their disease because the cells were completely resistant to all the chemotherapy that had been tried in that patient. So we wanted as tough a target as we could get, so we used this. And so let's start this up. We'll see the T cell come in. So you see the T cell connecting with the cancer cell. And what I want you to focus on is the lining of the cancer cell, all right, this outside part that's called the cell membrane. So see what happens when the T cell hooks up with the cancer cell. You see this bubbling that occurs. That bubbling is the destruction of the lining of the cancer cell. The T cell doesn't care that this cancer cell is resistant to chemotherapy. The cancer cell is not resistant to having holes poked in the membrane, and this cancer cell dies. The T cell lets loose, goes on to another cancer cell to kill it, and more importantly, that interaction between the T cell and the cancer cell causes the T cells to grow. And so you start out with a small number of T cells, and you end up with a huge number of T cells, and I think that's really, really important. So that's about how this works. So let's skip these two. So here's the process of how we make these cells. So we'll collect T cells from the blood of a patient, and what I'm showing you now is mostly been done on patients with leukemia because that was where everybody started because they knew how to target these cancer cells. And we take these T cells out of the patient, we bring them to a lab that's set up to create cells that can be used clinically. That's extremely important. We expose these T cells to a virus. So this is where it gets a little bit technical. So the virus is actually what changes the genetic makeup of the T cell. It engineers the T cell with a new gene that produces that CAR protein. The CAR protein then is actually produced by the T cell and ends up on the surface of the T cell. That drives an interaction between the T cell and the cancer cell on the bottom and hopefully kills the cancer cell, and I've shown you how that works. So this is about a 3-week process between collecting the cells, engineering the cells, growing the cells, and giving them back to the patient. It's not something that happens in a day. So that's about how we do this. Another big part of this is we need to grow the cells so that we start out with a small number of cells, end up with a larger number of cells in the lab, and then when they go into the patient, we create an even larger number of cells. And in order to achieve that, we use a system where these beads, these yellow ping pong balls, stick to the T cells, which are the kind of fried eggs in the middle here, and these provide all the activation signals the T cells need so they grow, 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 and are still very powerful, young, and active when they go into the patient so they can continue to grow in the patient. So this is just a picture of that, of how that cell culture system, the actual manufacturing system works. And almost all of the groups at the various hospitals, at the National Cancer Institute, at Chop and Penn, at Sloan Kettering, now at Seattle, have used some variant of this sort of way of growing T cells. So that seems to be at least one very good way to do that. So we've been doing this for almost 3 years now at Children's Hospital in Philadelphia, and between Chop and Penn, which is the university that we work with and where I'm faculty, over 130 patients have been treated with this specific T cell therapy, which targets this target that's called CD19 that happens to be on B-cell cancers, including ALL, the most common childhood cancer, non-Hodgkin's lymphoma, which is very common in adults, and a chronic leukemia that only happens in adults. And so in our experience, we've treated almost 40 patients, and these are patients who had refractory, in other words, their disease was not responding to anything. They had refractory ALL. They had actually, many of these patients had actually relapsed after a bone marrow transplant. And these were patients, unfortunately, for whom we have no good therapy. And so the first surprising thing that we noticed is that 92% of the patients that we've treated so far have gone into a complete response or complete remission. Now, complete remission is a step. It's not there all the way, because you can be in remission and come out of remission, and that's called a relapse. And there are patients who, after we treated them with these cells, did relapse. And some of these patients relapsed because their T cells didn't stick around long enough, and those patients we were able to retreat. And some of the patients, 5 of them, actually lost that target. The cancer cell was smart enough to get rid of the target, and then they were able to dodge the T cells, and then those patients we weren't able to retreat. We've been following these patients. The first patient that we treated was almost 3 years ago. Back in April of 2012 is when we treated her. She's actually at the White House today, visiting with the president at a personalized medicine summit coming out of the State of the Union address. So she's sort of become the international face of cell therapy. We have patients who are out a year or more. And I think most importantly to me, what I actually do clinically is I'm a bone marrow transplanter. And what I'm excited about is that only 3 of the patients that we've treated have actually needed a bone marrow transplant, and the rest of them haven't. So I'm very, very excited about the fact that the cells, at least in some of the patients, stick around. How long do they stick around? Well, this graph just shows you that. And if you're used to looking at like a graph like this, it basically shows the percent that keep the T cells over time, and basically 6 months out, about 2/3 of the patients still have their T cells. So that these engineered T cells do persist, they do stick around, and we hope they're still on the hunt. In terms of how the patients have done, about 3/4 of the patients are still living after 6 months after getting the treatment, but some of the patients have had their disease come back. And that's why it's not 100%. And it's absolutely not 100%. It's important to understand that a 92% complete response rate is not 100%. It is not 92% survival, because some patients can have their disease come back. So this messy slide, I really apologize for this. I just want to give you this sense of what happens in terms of the T cells proliferating, growing, expanding in the patient. I said that was important. And this is just a way of measuring that. And if you look at this graph, what all this graph is showing you is that all the patients who responded had cells that grew not only doubled or tripled, but 1,000, 10,000, 100,000 fold, 100,000 times more cells. So when these cells grow and do their thing, they have to grow a lot. And so we really do need cells that grow a lot. And then if you follow this graph out over time, you see some patients who keep their cells for a long time, a year or longer, and you see some patients who lose their cells a little bit more quickly. So that's that persistence part. There's at least some of these patients who actually hang on to their cells for a very long time. Now this is a complicated slide, but it really only makes one point. So some of these patients, all the patients get a little bit of chemotherapy before their T cells. Some of these patients have a response to that chemotherapy because their disease isn't completely refractory. And so if you have a pardon me, I'm sorry. If you have a small amount of disease in the bone marrow, which is where leukemia hangs out, our success rate is 88%. If you have a very large amount of leukemia, really no treatment is working at all, the complete response rate is still 82%. So even though there's a lot of disease, these T cells can find it and make it go away. There is toxicity. There's a treatment toxicity that's very different from chemotherapy. It's something that reminds me of stuff I see in transplant. It's called cytokine release syndrome. It's seen across all of these immunotherapy programs and platforms. Some patients can get quite sick. Some patients just get a fever and a headache. It just depends. Although I think if you look at the second point here, probably the most important thing is how much cancer there is, because the more the T cells have to grow to get on top of the cancer, the higher the risk of this complication is. We have learned a very important thing about what causes this, and we've been able to control the toxicity using another bizarrely named drug that's given to patients with rheumatoid arthritis. Who knew that you could use this in cancer? This drug called tocilizumab. The details don't matter, but I think the important thing is we've understood why the T cells can make some patients sick, and we have a treatment for those patients so that we can get them through the bad part and they can experience their complete response. Now, I want to briefly go on. So this is what the leukemia world looks like. There are a number of groups who've seen similar results. We're very excited about this. I mentioned the other hospitals that have been doing this, but the National Cancer Institute has started to apply engineered T cells to patients with a solid tumor, and that's really what we're looking for. That's the are we there yet? And so this is the same idea. The patients get chemotherapy to start with, and then they get these T cells once, and then we follow them over time. There was a specific kind of sarcoma these patients had called synovial sarcoma, and there were 11 patients treated, and some patients had very significant or even complete responses. So that's our first really good sign that these engineered T cells that grow like this can get on top of a solid tumor. And this is just gives you an example of what this looks like. I think we can just focus on the pictures all the way on the right. So this is what's called a PET scan, and all these black dots shouldn't be there, and they're all, unfortunately, cancer in this patient. So this patient had a lot of this particular sarcoma, was given T cells. A month later, a week to a month later, it looked like the patient actually might be sicker. There was concern that the cancer had gotten worse, but that wasn't what was going on at all. The T cells were attacking the cancer cells, and then 3 months out, you see the patient is in a complete response. So this is our first hint that this might actually work outside the leukemia field with large disease burdens, with a highly proliferative and active T cell. So I think, you know, if somebody asks me, is this going to work in solid tumor, these are the data that give me some hope that that might actually be the case. I won't get into this. This is just another example of a patient who had a large localized tumor, got these T cells, and then we see the tumor disappearing, nothing active now, still abnormal on the scan because the abnormality stays there, but no active cancer in this patient. So patient responded with a pretty large solid tumor mass. So again, are we there yet? Maybe not quite, but this is what it might look like. So in sarcomas, we're very excited for this particular kind of sarcoma. We do see some control of disease. They do see this cytokine release syndrome, the fever that I mentioned. Patients can be somewhat sick with that, but not terribly sick. And so, and again, I mentioned expansion and persistence. We see both of these, we being the group at the National Cancer Institute who originated this, especially Dr. Crystal Makel, who has really been the driver of this particular approach. Want to mention briefly that we're applying this to neuroblastoma, and this is work that was actually funded by Solving Kids Cancer. So again, we have developed a chimeric antigen receptor to engineer T cells using either an approach that permanently or temporarily modifies the T cells. This is just mouse data, but what you see, these pictures are mice with cancer. We're able to pick up because the cancer has been genetically engineered to emit light, and you can see control of disease with both permanently and temporarily modified T cells, which is the approach that we're going to be taking forward in a clinical trial. So what about neuroblastoma? Well, we know that this particular target, GD2, works for when we do an antibody, so that's very hopeful. It might work for other solid tumors like osteosarcoma. That's very hopeful. The group at Baylor has seen some complete responses. They haven't seen the cells grow in the same way that I was just describing to you. So I'll be very interested to see what happens when we have highly proliferative, very active T cells going after this target. We're starting this study, very much supported by Solving Kids Cancer, and we're starting with temporarily modified T cells where this CAR protein goes away, just to keep things as safe as possible, because the one thing I've learned from our prior study is that while the cells are very powerful, they also can cause side effects, and I want to be as careful about this as possible. I'm just very thankful that in the data that I showed you on that study that none of the patients who did have side effects, about a third of them had pretty significant side effects, none of those patients actually died of those side effects. So where are we? Well, I think we can control small amounts of disease, what we call MRD. But in patients with large amounts of disease in a relapse, I think at least in leukemia, we can get them back into remission. We can limit the amount of disease they have so they can go to transplant. And so a lot of people are touting cell therapy as a bridge to stem cell transplant. That's what I do. I'm a stem cell transplanter. But what I want to think about, oops, I missed a slide, so apologize, but what I want to think about is the notion about whether we can actually replace stem cell transplant with these cell therapies. And so that's what we want to look at, because a lot of the patients we've treated, at least with leukemia, never went on to transplant. And so is it possible? Yes. Is it sure that we can do this? Not at all. The thing I'm really excited about as well is that this particular cell therapy that I was talking about in leukemia, the first thing the drug company that has licensed this wants to do is to get an indication in pediatrics. This young lady all the way over on the right is that young lady that I mentioned who was the first patient that we treated. She's at a charity bike ride for our local cancer center. She was our top fundraiser at that ride, and I had a chance to do that ride with my daughter, who's on the left. That was actually a lot of fun. We hope that this will be a actual product that's available for physicians to prescribe to their patients as early as 2016. So they were pushing very hard to get to that point, and I think that really allows us, when we develop these programs for patients who have other diseases like solid tumors, as long as we can show some initial promise that this might work, there's going to be a lot of interest in adapting these approaches, even in pediatrics. Everybody complains, and quite rightly, that there's not enough focus on pediatric oncology, but I think if you can develop compelling data, people do listen. Want to mention, if you haven't read this book, it's a terrific book, The Emperor of All Maladies. It talks a little bit about the approach to developing cancer therapy, and I understand that PBS has created a Ken Burns documentary based on this book, and that will be showing at the end of March. So you might find that interesting. It's a great, the book itself, I can tell you, I don't know anything about the film, but the book is a terrific description about how everybody figured out how to treat cancer and where we got to where we are right now. And I'm going to end now with a incredible word of thanks to Solving Kids Cancer, who has been so supportive of this particular development program, who has really challenged us to get this up and going in patients with neuroblastoma, to focus on solid tumors and not just do leukemia that everybody has started with, myself included. I'm as guilty as anyone. So they've done a great job really providing leadership, and also it's important to note that they have worked with a lot of other foundations to bring funding to pediatric cancer, to solid tumors, to this particular innovative cell therapy. And for that, I really am quite appreciative, and now we'll take any questions.
Thanks, Dr. Grupp. Please submit any questions you have. We've asked people to submit them throughout the webinar, but if you've been saving them up, now's the time to submit them, and we'll go through as many as we can in the few minutes we have left. One question that we have already, Dr. Grupp, it's about neuroblastoma, and somebody that has a child with recurrent neuroblastoma that's been pre-treated with GD2 antibodies as well as natural killer cells, and they were wondering if that pre-treatment would have any effect on the T-cell treatment.
So I think that's a great question. There are two parts to that. One is, would it render the patient ineligible for the T-cell treatment? And the answer is no. If we had that rule, we couldn't treat anybody, because almost everybody gets a GD2-directed therapy now. So fortunately, that's not a concern. Would a treatment directed at either GD2 or with natural killer cells make a difference in the likelihood of success? Again, I don't think so, but I won't know until I actually give it a try. So it's hard for me to know whether it would make a difference, but it certainly wouldn't interfere with us being able to do it.
Okay. Following up on the pre-treatment for neuroblastoma, another participant wanting to know about previous radiation treatment and that having any negative effect on the enrichment of the T cells or the effectiveness of the T cell therapy.
Well, I think that's a great question. My sense is the answer to that is no. Prior radiation therapy, we've certainly had lots of leukemia kids that we've selected, we've been able to collect T cells on who've had prior radiation therapy, and it doesn't seem to make any difference whether you had radiation therapy in the past. I think getting radiation at the same time might be problematic, but that's not our intent, so I don't think that would be a problem. So again, I think I have data, at least in the leukemia world, that having gotten radiation beforehand, which of course is standard of care for the vast majority of kids with high-risk neuroblastoma, that shouldn't make any difference.
Okay. Another question is there a way to indicate in a screen when you're looking at a patient to engineer T cells for? The patients either have good or bad T cells that would make it successful?
Yeah, that's a fantastic question, and everybody sort of beats ourselves up with that question. I think the answer is we don't know. Certainly, patients who have been aggressively treated with high-dose chemotherapy over and over and over again don't have any T cells that we can detect in the body. And those patients, I'll be honest with you, we don't even try to collect T cells if we can't measure any T cells. So we know that's not good. However, most chemotherapy for neuroblastoma, especially most relapse treatment for neuroblastoma, don't leave you in that situation. So we're very hopeful that at least from a numbers standpoint, the T cells will be okay in those patients. We think it's actually easier in the solid tumor patients than it is in the leukemia patients because of the nature of the kind of treatment that the leukemia patients get. So I would say actually we're very hopeful from a numbers standpoint that we're going to do better in terms of being able to collect T cells in solid tumor patients, specifically neuroblastoma patients. However, there are patients, I mean, when I said that the complete response rate was 92%, what happened to the patients that didn't respond? Well, what happened to those patients is that even though the T cells grew in the test tube in the lab to create the T cells for the patients, the T cells didn't grow the way we wanted to in the patient. And I've shown you very clearly that in order to have the really strong responses that we're talking about, those T cells have to grow. So we have studied this, and we don't have a good answer, but we're continuing to study this. What are the characteristics of T cells if you have an okay number, but they just don't grow the way that you want? And can we convert any of those patients who have T cells that don't grow very well into patients whose T cells grow better? So the answer to that question is that we certainly know about the numbers. We know the appropriate number of T cells to collect. It's not a high number. It's not a normal number, but just some T cells is probably enough. But there are some patients whose T cells just don't like to grow. It certainly correlates with having a great deal of chemotherapy before the T cells are collected, but we don't have a real sense for when the cells just refuse to grow why that might be.
Okay. Another family asking about T-cell therapy. It sounds like they have a unique situation. The child's in remission for neuroblastoma, but then also has therapy-induced MDS. And the two-parter is CD19 expressed on either of those, neuroblastoma or MDS? And the second part is, if a child has more than one malignancy, can T-cell therapy hit both malignancies?
Well, so the first question is that CD19 is not expressed on either neuroblastoma or MDS. MDS is a different kind of cancer. CD19 is really expressed on lymphoma and leukemia of B-cell origin, and so unfortunately the answer to that is no. The answer of more than one cancer would be an answer of more than one target. So if in principle, if we had more than one target, but we had T cells that could recognize both targets, you could give them together. There's a lot of work in the idea of combining, a T cell that recognizes one thing with a T cell that recognizes another. So in theory, that would be possible. The hard part, and I hate to give this answer, but the hard part is that the myeloid type, AML, MDS, those sorts of things, those malignancies, we are working on targeted treatments for that situation, but it's a little bit harder because the targets that we would choose would probably be on normal bone marrow cells. I think we can get around that, but it's why that we've been waiting actually until this year to give that a try.
Okay. The next question is about patient that would be already gone into a complete response or an NED status, and wondering if the T-cell therapy could be used as a maintenance therapy?
Yeah, that's a great question because a lot of the focus in neuroblastoma has been on treatment in either with minimal residual disease or no detectable disease, which is to say remission. And that's how Accutane was developed. That's how immunotherapy is best used. And so it makes a ton of sense to think about that. Right now, the answer would be we wouldn't do that. And there are several reasons why. One is that the regulatory agencies like the Food and Drug Administration want you to be able to say what the cells do to the cancer, and the only way to have that is have detectable cancer. And then the other reason is that until we understand the risks of doing this better, it would personally, for me, be hard to justify doing this if the patient didn't have active cancer, because everything we do on a clinical trial does involve some risk. Now, if this works well and it's well tolerated, then of course the people who are interested in, we're all pediatricians, we all believe in prevention, we don't want to treat a relapse, we want to keep a relapse from happening, then that would be the next step. But I would say that the bar for that is way higher than where we are right now, because we really have to show excellent safety before we consider something that's used in the remission setting. I don't think it's impossible, but it's not the first step.
Let's switch up to talking about another type of high-risk solid tumor. There's a family who has a child with a relapsed Ewing sarcoma in the lungs and wants to know the trials that you reference, are they specific to neuroblastoma, or are other sarcoma cases being considered as well?
So the National Cancer Institute trial is very specific to only one kind of sarcoma. Which is the synovial sarcoma, which is not the same thing as Ewing's. There are a small number of Ewing's patients that have the same target, and one of the discussions that I've been having with the company that has licensed their technology is, can we get past the specific sarcoma and start testing it in patients who have the target regardless of what their tissue diagnosis is. And I'm hopeful that in the future that they'll be going for that, but right now they're just targeting the one particular tissue diagnosis. And that's a blind spot for us cancer doctors. We've always been used to designing trials for specific diseases, but now in the era of targeted therapy, I think the question we ought to be asking is, do they have the target? Does the patient's cancer have the target? And if the patient's cancer has the target, we ought to try the treatment. So the answer for right now is we don't have anything for Ewing sarcoma. I would hope that in the near future, near being in the next year or so, so not that near, that we can at least expand this particular cell therapy to other patients who have the target but not the same named diagnosis. And the other thing I would say for the further future is that there's a large group of doctors who have gotten together trying to find other targets for these immunotherapies for pediatric cancers, and Ewing's is very high on the list because of the lack of really good therapeutic options for patients whose diseases have come back. So I think there's a lot of focus on this. People recognize it's sort of an orphan circumstance and that we really need to pay attention to it. There's been some significant funding applied to target discovery, but having something right now, unfortunately, there's not.
Okay. Moving on to another cancer type. Someone wants to know, would this type of therapy work for pediatric brain tumors? And then I'll just ask, what work is being done in that area clinically?
Right. I didn't actually have enough time to get into that, but there is a lot of work that's happening with both adult and pediatric brain tumors using engineered T-cell therapy. The group at Baylor has really been leading the way against some pediatric brain tumors. We do have potential targets that could be used. There are initial brain tumor trials opening up, and actually the group through Stand Up to Cancer that I mentioned that does pediatric target discovery is one of the trials that they're doing. In addition to the leukemia trial, which I just described, they're also doing a trial in pediatric brain tumors, and that's being led by the spearheaded by the group at Baylor. So there's very much a lot of interest in that. Again, are we there yet? We're absolutely not there yet. This is just beginning. People are just starting to see how this might work, and we every time now that we have such powerful cells, I think what everybody wants to do is they want to move into this very carefully. because we recognize that the risk of side effects is there. And brain tumors being in the brain, a significant amount of inflammation caused by even a powerful and effective anti-tumor response might cause the patient to get sick or have significant risk. And so we're definitely headed in that direction. People understand this is a field that's crying out for innovation. We very much want to do it, but we're trying to do it as safely as possible, just because brain tumors are in the brain.
Okay. A couple technical questions. One is related to currently the natural killer cells in certain clinical trials are being used therapeutically that are donated from the parent. And they wanted to know if in this case, using T cells and engineering them, could a parent donate those cells?
So the answer is that would not be possible unless the patient had already had a bone marrow transplant from that parent, which is possible. I've seen that happen. The issue is that the T cells would there's a complication of bone marrow transplant or stem cell transplant that's called graft versus host disease, and that's basically an immune attack of the incoming T cells against the patient. And basically, the risk of graft versus host disease would be very high in that situation, and so there's not a good way to use the parent as the donor. And the worry about graft versus host disease is why I, in the clinical trials that I've done, have really focused on using the cells directly from the patient. And NK cells, by the way, just to answer that part of it, NK cells don't have that risk, and you can cross these barriers very nicely with NK cells, which is one reason why I'm a T cell guy. I'm not an NK cell guy, but I really appreciate the NK community because these are cells that can kill cancer cells in the test tube. There's no doubt, and there's no risk of this sort of reaction.
Just being conscious of the time, we've got a couple more questions, Dr. Grupp, if you have a few minutes. So
absolutely, absolutely.
Another question that's somewhat related to the previous is about stem cells that are banked for the child and maybe left over, and the person wanted to know if those could be engineered and turned into therapeutic T cells.
Just a moment, pardon me. So there are people who are trying to do just that. In theory, there's no reason in the world why we couldn't do it. I personally have not tried it, but the group at MD Anderson has used cord blood T cells. I'm not sure if we're talking about stem cells or cord blood, so let me separate them both. So cord blood, which was banked at birth in the child, intrinsically there's a reasonable chance that you could make engineered T cells out of that. We haven't done it, but the MD Anderson group. If we're really talking about stem cells that were collected at the time of preparing for a stem cell transplant for that individual patient, the answer to that is that we have not had good success with trying that. Basically, the medicine that's used to collect the stem cells, which is G-CSF or Neupogen, doesn't do a good job for the T cells, and those T cells don't grow very well. So we've talked about kind of revisiting that question because, of course, having something in the freezer and not having to do the cell collection procedure would be much easier. See if we can get these cells to grow a little bit better, and there's some process development around that. But right now, I think that being on Neupogen at the time that the stem cells are collected is not good for your T cells, and we haven't gotten the T cells to grow. That's an area of research, but right now we can't use it.
Okay. Question about neuroblastoma. They've heard that neuroblastoma creates its own environment that can make it resistant to T cell therapy, and wanted to know if you could lend your perspective on that.
So that's a fantastic question, and the whole issue of are we there yet in solid tumors is about that issue. So in addition to dodging the immune system by downregulating targets, the other thing that solid tumors learn how to do, probably much better than leukemia, is to create an environment in the interior of the tumor that excludes immune cells. So the answer to the question is that I think the bar is higher for solid tumors. However, these are if the T cells are proliferative, if they're powerful, if they cause a lot of inflammation as these T cells do, I think there's reason to believe that that might be the case. It's also true for sarcomas that they can have immunosuppressive interiors in the same way, and as you can see, we can see some evidence of disease control in even with patients with fairly large tumors. So I think when we're all very excited about the solid tumor field, we also are fully cognizant of the fact that solid tumors do have mechanisms to avoid the immune system. We hope that highly active and proliferative T cells are the way around that, but that's why we have to do these clinical trials. So that's, you know, the person who asked that question really has put their finger on the key concern and why we have to sort of try this and see how it's going to work.
Got a specific question, again, in neuroblastoma, and this is about the potential benefit of T-cell therapy in a CNS or brain relapse in neuroblastoma.
Yeah, that's a great question. I mean, the answer is I don't know. However, I have some hope for that possibility, and let me describe why. So I talked about the leukemia study, and ALL, the leukemia that we were studying, also can go to the brain, it can go to the spinal fluid. And so when we started treating these patients, we said we wouldn't treat anybody with a lot of disease in the spinal fluid, but we've treated a couple patients who had a small amount of disease in the spinal fluid. And so the two things I can say is that almost all of the patients we've treated, we see T cells in the spinal fluid, which is amazing to me. So the T cells actually get in there. I don't know how they get there, to be honest with you, but you can definitely see them easily. So they can go there. Now, are they going to do the trick? Well, the two or three patients, actually two, that we treated with small amounts of CNS leukemia did go into remission with the T cells. So maybe. The answer is maybe. And so I think that all of these programs are really, really cautious about CNS disease because of the extra added possibility for toxicity. But I can tell you that as soon as we got enough experience with patients without CNS disease, we've now opened the trial up to patients with CNS disease, and we'll learn, at least in leukemia, whether that's safe or not. And if it's safe in leukemia, that'll give us an opportunity, if these cells are working for patients with neuroblastoma, to try that in the future. It's not the first step, but we're recognizing that the CNS is actually an important sanctuary for patients with neuroblastoma. You know, 15 years ago that wasn't so obvious, but it's definitely true now. So I would say there's hope, but not certainty in that regard.
Okay. A couple questions came in. How are T cells working post a double stem cell transplant for neuroblastoma? And have you had a good response with your clinical trial?
So we haven't treated anybody like the clinical trial is just opening, so the answer to that question is we don't know. I think if you have adequate T cells, and I'm probably the guy who's as responsible for the tandem stem cell concept as anybody in the world. I think that if you have, if you're a month or two out from a tandem stem cell transplant, probably those T cells are not in a position that they're not going to be enough of them to actually be able to do anything. But I think patients who are a number of months out, 6 months out, we've studied T-cell recovery in those patients after tandem stem cell transplant, and they have perfectly adequate T-cell recovery at that point. So my sense is that that wouldn't be a reason for it not to work.
Okay. And these last couple of questions are really about the future and what that potentially looks like. First one is that in considering combination therapies, do you see this approach possibly used in conjunction with some of the emerging precision medicine trials that are up and coming?
So the answer to that is absolutely. Especially in solid tumors. If there are drugs, for instance, immunotherapy drugs called checkpoint inhibitors that might be helpful in getting T cells to overcome that immunosuppressive immune inactivating environment of the cancer. So if that's a limitation, then combining cell therapy with checkpoint inhibitors might be the way to go. And lots of people, including our group, have started to design trials around that. So I think there's a huge amount of interest in that.
Okay, and lastly, there's a question that wants to ask about some of the goals for 2016 related to solid tumors and brain tumors, maybe for your own lab, and then if you could expand upon for the field in general.
Well, it's the beginning of 2015, so what I hope is that several groups, ours included, have a initial feasibility and some efficacy data in several pediatric solid tumors, neuroblastoma being the first, but hopefully brain tumors as well. I hope that we can adapt the NY-ESO, the synovial sarcoma approach, to other pediatric solid tumors that express that particular target. And then my great hope is that the Stand Up to Cancer initiative will find other targets that might be useful in pediatric solid tumors. And the reality is that if we can find a good target, we can get to an engineered T-cell very quickly. We have that pipeline to create the engineered T-cell very quickly. So the hard part is finding the target, and there are now dozens of doctors involved in looking for them. So for 2016, maybe we found another target or two that will allow us to expand this engineered cell therapy approach to other patients.
That's great. Well, Dr. Grupp, all of us here want to say thank you for spending a lot of time on the questions and answers, which is important after providing a lot of this information, some of which is new for the first time. And I also want to thank everybody for joining us today. And we realize that there's a lot of information that was covered, and so if there are any other questions that come up in the next few days or anytime later, please feel free to forward them to us via email, and we'll get them all answered. The email address to send any follow-ups to would be info@solvingkidscancer.org. And I mentioned before we're going to follow up with everybody with an email link to the archive of today's webinar. That'll happen in the next few days. As well, we ask you to stay tuned for an announcement of the next webinar in the series that will take place sometime this spring. So thank you, everyone, again. Thank you, Dr. Grupp, especially. And I want to wish everyone a great weekend, and we'll look to see you again in the spring for another webinar.
It was my pleasure. Best of luck to everybody.